I'm probably not going to get through all the slides, but as I said, they're all available on Panopto. So my recordings from last year are still online. So please take the opportunity to look at those, because we've only got about 45 minutes left of this session. So in the remaining time, we're going to talk about drug design approaches and mechanisms by which we can design drugs. And if you were in the Frontiers module earlier in the term a couple of weeks ago, you'll be familiar with some of this because again we go through this sort of dichotomy between structurally based approaches and functionally based approaches. So the approach based on knowing what the structure of something is and knowing what structures can interact with the structure of our drug target, and going from a functional based approach, which is a screening approach where we don't really care what the structure is that we're trying to target. All we're doing is we're testing lots and lots of different things in order to see whether they work. So we're going to be talking about both of these because both of them are used in drug design, and frequently they'll be used at the same time. They'll be used in combination with each other. So the first one of these is rational drug design. So rational drug design is essentially if we understand exactly the molecule that we're interacting with. So obviously we need to know an awful lot about the disease state. So if we have a receptor in the membrane, we know what the shape it is, and we know there are various charges on it, we can start to think about, well, what is our drug molecule going to look like in terms of its shape, in terms of its size, in terms of its charge, its physicochemical properties, stuff like that. So for fully rational drug design, we can essentially go from knowing everything there is to know about a receptor or an enzyme or transport protein and then designing a drug specifically to fit. So our drug molecule here would be exactly the size and shape to fit into the active site, and it would have the charges and the hydrogen bonds and the van der Waals interactions and all that sort of stuff. Remember that most drugs are going to interact with their target molecules through a series of non-covalent interactions. So it's not going to be strong covalent chemistry, but through the multiplicity of these non-covalent interactions, we do get a strong interaction. So the affinity of interaction is usually very high with drug molecules. So this fully rational design may be described as a process of developing a full understanding of the 3D structure of the target and then selecting or optimizing or designing a compound to interact with it. Now, instead of usually just going through one cycle and saying, right, here is our target molecule, here is our drug molecule, let's see if they interact, then usually there's a cyclical process. So we'll go around this cycle a number of different times. So through the target generation process, and remember this is the bit that can take an awfully long time, this is the bit where we decide what our target is for our drug molecule. We come up with the idea of a drug target, so the enzyme or the receptor or the transport protein or whatever that we need to inhibit, and we know a bit about it. So we need to know something about it, we need to know what it is. And we will either have or we will generate, or a combination of two, of both, structural information about the active site of the enzyme or the receptor or whatever. So the business end, the bit we're going to try and target. If we have that information, we'll use it. We'll probably need to develop more of information about it to generate that information in sufficient spatial resolution, sufficient chemical information resolution. We then design our inhibitor molecule, so we design the molecule which is going to interact with our drug target. And then we will typically put them together and see what happens. So we'll put them together and see whether or not they interact. Now this will be in the lab, this will be in vitro, this will be in a test tube or a plastic tube or whatever. So we will purify the drug target and we will make it in the lab. We will make the inhibitor molecule, we'll put them together, we will determine how they interact. And it may well be that we'll do some binding kinetics, we'll determine how much of the inhibitor is bound and how much is unbound. So we'll be able to develop isotherm models, we'll be able to look at the binding affinity, we'll look to see if there's cooperative binding, if there's one molecule which binds per drug target or more. We'll look sort of to see what will prevent binding from happening, to see whether or not binding happens in all conditions, at all pHs, with solute concentrations, with different solvents, stuff like that. So the objective here is to understand how the molecules interact. Because if this is what's going to be happening inside the body, we need to be sure that once we've made this and turned it into a drug and given it to someone, it will happen in the body. And it may well be that our inhibitor and our target molecule interact really closely and strongly in the lab at 50 degrees centigrade and at a salt concentration of 10 molar and various other things. But we have to think about conditions within the body. So we have to think about exactly how this interaction would proceed. So we then generate from this process an awful lot of information. So even if we know stuff about the drug target molecule, we know stuff about the inhibitor molecule, we can still generate more information from going through this process. So we solve the structure of the drug target or map the mechanism of action, because remember, even if the drug target molecule is relatively well characterized, even if the structure is known, we still probably need to know how it actually works. So we need to know about the structure-function interrelationships. Designing our inhibitor molecule will require us that we know something about molecular interactions, so we know about how the drug interacts with our drug target. And clearly, when we design our inhibitor molecule, as I said, it needs to be binding and interacting with the target in physiological states, so under physiological conditions, but also it can't be toxic or have side effects. So this is a good time. So rational design is a good time to think about how much of a drug-like molecule our drug target, our drug candidate molecules actually are. So if we're designing our inhibitor molecule from scratch, we can start to think about things like, well, we want to make sure that this isn't toxic. We want to make sure that this is going to be available, bioavailable. It's going to be absorbed well. It's not going to sequester itself into a certain body mass. It's going to actually get to the site in the body that it needs to get to. So rational design, the concept behind rational design is a very good one, because it taps into this idea that we will understand the interaction and the interplay between structure and function. And potentially we can quantify that. And this brings us into sort of QSAR, so quantitative structural activity relationships, which is there's a slide on later in the talk. The protein structure determination is needed for rational design. This is usually in the form of X-ray crystallography or NMR. So in order to work out what our drug target molecule looks like, that will either already be solved or we will need to solve it. The tools for that are X-ray crystallography or NMR. X-ray crystallography is the gold standard because the resolution is incredibly high. So the resolution is sub-angstrom, if you're lucky. It is unfortunately a very difficult technique. So you have to purify your protein, you have to then crystallize it. So what we have on the top left here is a picture of protein crystals. This is very much a dark art, making protein crystals. You have to essentially purify your protein, you put it in lots of different buffers, and you then set those buffers up in lots of different conditions, and then you wait until crystals form. And it could take a week, it could take a month, it could never happen. So it's as I say, it's very much still a dark art. We don't really understand exactly if it's going to work or not. Once we have a crystal, a nice crystal that looks a bit like that, we then subject it to X-rays, and then we get a crystallography pattern on the right-hand side. That's the diffraction pattern, and then it's a case of solving the structure from that pattern, which is a sort of time-consuming, laborious, and mathematically complex process. But you end up with a molecule that looks a bit like this. So you end up with a solved structure. And as I said, you can get this down to the sub-angstrom level, which is potentially very useful, and that will give you information about the structure of the molecule, and also it will give you information about the chemistry of the molecule, because you know the protein sequence, you know which amino acids are in which position, so you can then assign different amino acids to different portions of the molecule. So that will then tell you exactly where positive, negative charges are, and aliphatic groups, polar groups, and stuff like that. The difficulty with X-ray crystallography is the crystallization step. It's relatively straightforward for soluble proteins, things like enzymes. For membrane proteins, receptors, it's a lot harder. So most receptors you can't crystallize because it's very difficult to crystallize something in a membrane. So you have to purify your protein. Most receptors will not purify without most receptors will not fold into the correct shape without the membrane being present. Without the membrane present, basically you've got no receptor. So it's very difficult. An alternative is NMR. So NMR, nuclear magnetic resonance. So NMR can be used to generate protein structures, but the resolution tends to be lower. NMR can be done in solution, so you can do NMR determinations more readily of membrane proteins, but as I said, the resolution tends to be a bit lower. We do have NMR kit on campus here, which is very, very powerful and can get up to a high resolution, but the gold standard is still X-ray crystallography. And as I'd alluded to, analysing the structure of the protein-ligand complex is also very useful in rational drug design. So we've got our target protein structure, and we've got our protein-ligand complex. So we can utilise all of these bits of information in order to expand our knowledge area and our knowledge space within the field. So this can be very useful for development of new drug molecules. It can be used to diversify drug libraries and things like this. So if we start with our target protein structure, this would be our enzyme or our receptor. We will then use computation usually in order to design our ligands, so design our drug candidates, and we will synthesize that using organic chemistry. We will usually biochemically evaluate that before it gets near our drug target molecule, so that will be the biochemical evaluation will be things like toxicity, things like drug-like nature, solubility, stuff like that. And then we've got marrying the drug candidate with our drug target molecule and determining the structure of the protein-ligand complex. So again, this will tell us how the drug molecule and the target molecule interact, what sort of non-covalent interactions are present. We can make that a more thorough investigation, and this is often done using site-directed mutagenesis. So site-directed mutagenesis is where you take your drug target molecule and you change the structure of it. So you change the amino acids in order to test certain things out. So it could well be that we have our drug target molecule and we believe that the hypothesis would be that the way in which the drug and our target molecule interact is mainly through the charged residues. So we might want to knock the charged residues out. So if we knock those out, we change the protein, change the charged residues to similarly shaped residues which are non-charged, then we would then be able to test and determine whether or not we have similar interaction between the drug and the target molecule without the charged residues. And again, that would give us more information. It may well be that without the charged residues, we get a better interaction, a more physiological interaction. And then we sort of go around this cycle and iteratively improve the process. So if we start with one or a series of potential drug molecules, we would then change the drug molecule. Perhaps we'll add extra charges, perhaps we'll add extra side groups in order to give rise to a better bioavailability, better absorption kinetics. And then by the end of the process, we'll have gone around the cycle several times and we'll have developed a molecule which is more druggable, more useful. So this really is this idea of rational drug design, as I've said, relies upon knowing about the drug target molecule. And this kind of brings us on to the question of how many new drugs are really truly innovative. So how many brand new drugs are there per year? And the answer is unfortunately not many. So this is a real problem for the drug industry at the moment. So the majority of new drugs target known drug target molecules or their variants of existing drugs. And this is because it's easy. So this idea of low-hanging fruit, it's a lot easier for a company to target something which will make them money, but is not going to require as much investment, as much research in order to actually develop. So if you're thinking about statins, then a lot of companies make thinking about beta blockers, a lot of companies make beta blockers. It will be easier and quicker and cheaper and more cost-effective to just develop a better beta blocker than it would be to develop a drug to a brand new target molecule. So a lot of drug companies will just go along the same path and they will develop new variants of their existing drugs. So if you were to develop a brand new drug, you'd have to start a lot further up the development pathway. You'd have to restart and you'd have to identify what the drug target was in the first place. So pharmaceutical companies need to do something about this. So the regulatory authority, the FDA, the European Medicines and Healthcare Agency have commented about this an awful lot, and the drug companies are urged to develop new drugs, to new target molecules. The problem, of course, is that the drug product development process is incredibly expensive. Early steps can often lead to no lead molecules. So there's a very high failure rate, and it is a high-risk approach. So you will have seen in the pipeline yesterday that we start with a huge number of candidate drugs, and only a very small number of those will get down to clinical trials. So there's an awful lot of wastage. It could well be that a company will develop 100 potential new drugs and 2 or 3 of them would get to clinical trial. If those 2 or 3 fail in clinical trial, then the whole process has been for nothing. So there's an awful lot of cost involved with no potential reward. And so the sort of counter-argument to that commercially is that customers will only pay really high prices for really innovative drugs, not generics, not biosimilars or similar drugs. So there is a potential benefit from developing new drugs in that people will pay top dollar for them, but obviously there's a much higher risk. Now, one thing that has been done to help this process out is this whole area called orphan drugs. So orphan drugs are drugs which are developed for diseases which affect a very small number of people. So let's say 10,000 people in the USA are affected by a certain disease. So that would come under the heading of an orphan drug. So orphan drugs are looked at from a regulatory perspective in a different way. There's a lighter touch, which makes it easier to develop drugs for these orphan drugs, for these so-called orphan diseases. The result is that orphan drugs cost absolute fortune. So per dose, orphan drugs cost hundreds of thousands of dollars. And that is because they are innovative, but it's an example of where innovative drugs will give rise to a higher price, a higher cost. I put at the bottom here, advances in molecular biology and bioinformatics will help improve the situation. I'm not entirely sure whether that's still true, because molecular biology and bioinformatics have been around for an awfully long time now, so there have been a lot of very useful tools around for a long time. Drug development doesn't seem to be getting much more innovative. There is still an awful lot of inertia in the industry. There's an awful lot of inertia in terms of production of drugs and in terms of development of drugs, mainly because of the massive cost involved, the massive cost of changing things in these companies. Most drug companies are very large companies. And of course, there's a lot of inertia brought about by the regulatory authorities. So the FDA, EHMA are both have an awful lot of regulations involved with them, which we'll hear about on Thursday morning. So there are lots of barriers to innovation within the drug companies. So having said that, we've talked about the rational design approaches. So now we're going to talk about the alternative to rational design, which is screening. So if we've got a screening approach, it could well be that we know that we have a receptor molecule, and we know that our receptor gives rise to a disease phenotype. And we know that we want to inhibit the receptor. But we don't know what the receptor looks like. We know what the gene is, we know what the protein is, so we can purify it, but we can't solve it structurally. We don't know what it looks like. We don't know much about its physicochemistry. All we know is that it causes a disease, and if we knock it out, we can potentially stop the disease from happening. So then we'd use a screening approach. So what we would do is we would have the receptor, we'd have some sort of test. So in the lab, we would have some sort of test where this would give rise to a colour or a fluorescence or a light or something like that, so we could measure it. And then we would try lots of different things, lots of different compounds. So this is by far and away the most common route for drug development industrially. So the idea of screening lots and lots and lots of things. So this could be 1,000, it could be 100,000 different compounds. So this would have to be an assay which worked in fairly high throughput. And in fact, many companies pride themselves on their compound collections. So if you go to most big drug companies, they will all have a compound bank, and the compound bank will be in a big temperature-controlled warehouse. It will be robotically arrayed. It will be in fridges, and there will be 10,000, 100,000, 1 million compounds there, all ready to go and be tested. So many companies rely on this approach almost entirely. So in order to do this screen, you can use a compound library. You can also generate a compound library using what is called combinatorial chemistry. So combinatorial chemistry is something that it's a chemistry approach which is quite different to traditional chemistry approaches. So in a traditional chemistry approach, you have two compounds, so A plus B go to C. So you've got two substrates and one product. In a combinatorial chemistry approach, you'll have lots of A's, lots of B's, and then you will combine them. So if you have 100 variants of A and 100 variants of B, you combine them randomly together, and therefore you'll get 10,000 variants of C in different combinations of A and B. So the whole idea of combinatorial chemistry is that you can generate a very wide diversity. So combinatorial chemistry has a lot of impacts in terms of lead discovery and lead optimization. So you can use combinatorial chemistry to generate a large number of compounds that you can then test using a screening route. You can also use it to optimize leads. So if you know you've got your drug molecule, it has a certain shape, you know, let's say we have a drug molecule which is a heterocycle that looks like this, and it's got a sulfhydryl. So there's our drug molecule, and we want to optimize it. So how could we optimize it? Well, we might want to say, well, what if we change this bit here? And what if we add things here? And what if we add a bit here? And what if we fluorinate here? And what if we do all of that at the same time? So if we're interested here and here and here, and we've got, let's say, 10 different things we can put there, 10 different things we can put there, 10 different things we can put there, and 10 different variants of fluorine we can put there. Let's do all of that together at the same time. We'll generate a library of 10 to the 4 compounds, all with different combinations, and we can test which is the best. So we've already worked out what our drug is basically going to look like, but then we can optimize it. We can make it better by changing lots of things at the same time and then testing using our screening approach. So this combinatorial chemistry is an automated technology for producing large numbers of diverse chemical compounds rapidly. And these collections of compounds are called combinatorial libraries. So the basic concept, as I've said, is that simple chemical subunits are joined together to other chemical subunits in potentially every possible combination, and I've already talked about that. The big advantage of combinatorial chemistry is, well, one big advantage is you can make a very large number of compounds. Another advantage is it will be typically cheaper per compound. So combinatorial methods can be used to produce either discrete compounds or mixtures, depending on the format that you go for. So you can either decide to make 10,000 variants of this molecule, and so you'll have 10,000 reactions. So you'll have each reaction in a separate pot, and you do that robotically, or you can make all 10,000 in one go, in one vessel. So mixtures are very useful in the discovery of compounds with particular biological activities, for example, in drug discovery, but it is necessary to have a means of distinguishing them, the active compound from all the others. So if you've got 10,000 compounds in a pot, then you need to know which one is doing the business, which one is actually working. So there are various methodologies for combinatorial chemistry, and what we're going to talk about is solid phase synthesis. So solid phase synthesis is the one that is most typically used. So solid phase synthesis is essentially you have a solid phase, so you'll either have a bead or you'll have a pin or something like that, and then you will generate your molecules on the surface of that bead or that pin. The big advantage of using a solid phase is that it's relatively easy to separate. So with a bead, you can take it from one place to another fairly easily. You can certainly manipulate a bead, and this will be a bead that's, let's say, 5 millimeters across, or a similar sort of size. A pin would be sort of that long, a metal pin that you would then be able to dip into different reactions. The big advantage of that is that you can move it around. You don't have to rely on the fact you're moving molecules around, which is quite difficult because they're quite small. So if you attach the molecules to something big, you can physically move them around. It is possible to do this using solution or liquid phase synthesis, or with biological methods, which I'll talk about in a little while, but most of this is done on solid phase. So there are a couple of methodologies used when we use resin beads, because resin beads are a very commonly used method, and these are called the teabag method and the mixed split approach. And as I said, we'll usually use robotics in order to allow this sort of thing to happen. So over the next couple of slides, we're going into a bit more detail about solid phase synthesis in terms of resin beads. So The synthesis of these combinatorial libraries will usually use this solid phase chemistry. The compounds will be synthesized on some sort of solid support, and frequently these will be small spherical resin beads. So these will be a few millimeters across, as I've said. Because they're easy to handle, they're easy to take from one place to another, they're easy to sort. The reaction will be forced to completion by addition of excess reagents and reactants. So essentially you'll have all your beads in one pot, and you'll make sure all of the beads react 100% because you will have flooded them with reactants. So you'll take everything to completion. Now, this is one of the disadvantages of combinatorial chemistry. It is slightly wasteful in terms of reactants because you want to make sure everything proceeds to completion. Unwanted materials are readily removed by a simple filtration and wash step. So we can imagine what this is going to look like. We have our beads in a pot. We put our beads into a reaction vessel, which will be a fairly small reaction vessel. We'll add our reactants. We'll allow the reaction to proceed under whatever conditions it requires, so whatever temperature, pressure, solvents, stuff like that. When it's finished, we'll just filter them out. We'll just use a filter like a sieve. We'll take the beads out, we'll wash them, and then we can use them in the next stage. The origins of this method were in the 60s. So the origins of this method were in synthetic peptide libraries. And the example of the chemistry we've got here is one for peptide libraries. There are a lot of chemistries out there. I could do 2 hours on solid phase chemistries, but it would be quite a dull lecture. So if you're interested in this, you can go away and read about the myriad different combinatorial chemistries available out there. The original work, so this is a figure from the original paper, so Merrifield 63. So he was interested in generating peptide libraries. And he developed these on polystyrene beads. So he derivatized the polystyrene beads with this nitrosylated benzene with this chloromethane group on the end. And then he was able to stepwise build on peptide chains. So the way in which this is done, first of all, it's done the opposite way to the biological method. So the biological method of making peptides is from N-terminal to C-terminal, as we all should hopefully know. The chemical method is the opposite. The chemical method, you lay down the C-terminal first and then you move towards the N-terminal. You will essentially, each step is, the first step is the generation of putting the first amino acid on. The first amino acid is protected. Because this is all chemically done, it's not biologically done, because it's chemically done, we need to make sure the coupling reactions are done in a directed way. So there are no enzymes to help us here. Enzymes are very good because they put the right thing in the right place. Chemically catalyzed reactions are not so good because if there's a reaction, it will happen within whichever groups that kinetics dictate. So essentially, we have to make sure only the right reactions can occur at the right time. So in order to make a synthetic peptide that looks like this, one would start with the amino acid at the C-terminal, as I've said. So here's the amino acid at the C-terminal. It's got R group 3. It's got and it's got both N and C termini protected. So these are protected so they can't react. So what we do is we deprotect the N-terminal. This deprotection will be done using a chemistry, and then we will couple. So we'll deprotect in one step, then we will couple to our amino acid number 2. And as you can see, amino acid number 2 is protected at the N-terminal and it's unprotected at the C-terminal. So the C-terminal of number 2 will interact with the N-terminal of number 3. So we will get a peptide bond forming. This would be a chemically synthesized peptide bond. It's to all intents and purposes, it's the same as a biologically generated peptide bond, except that it's chemically catalyzed. So the reason we want to protect the N-terminal here and the C-terminal here is that if these were unprotected, then this N could interact with this C and form the amino acid chain in the wrong order. So this is why chemical synthesis of peptides is somewhat more complex. We will then deprotect number 2, and then we will couple number 3, and we will cleave then from the support, giving rise to our peptide. Now, as you can see, this is for a tripeptide. This has been an awful lot of work to make a tripeptide because we have to go through lots of consecutive deprotection, washing, coupling, washing, deprotection cycles. So this is not a terribly easy thing to do, but this is a method which is fairly commonly used industrially, and it can be done robotically, so it can be automated. It's also worth reminding ourselves that the R groups probably need to be protected as well. So if we have an R group that contains a carboxylic acid or a amine group, so if we have an acidic or basic amino acid, then the R group would have to be protected as well. Otherwise, the R group would also interact and react to form a peptide with a very weird bent backbone. Example protection chemistries are here. So commonly used protection chemistries are FMOC and BOC. So FMOC looks like this. It's a pyridine type chemistry, and BOC is this TFA group. So BOC is looks like that. So these will both be attached to the N-terminal, and then there will be specific chemistries, fairly harsh chemistries, that will be used to deprotect, so to take these off. And if, again, we look at the original paper, we can see the protection that was used. So a carbazonium was used to protect the N group of the first amino acid. This was then removed using bromination, and then the second amino acid, when it was added, was also carbonobenzonated. So we can look at the chemistry, and as I've said, there are plenty of references out there for different types of chemistry. And it's worth noting that this doesn't just work for peptides. So the example I've given here has been peptides. There are lots of chemistries where you're not using peptides. So our example of our drug molecule here, clearly you wouldn't use peptide chemistries here because this isn't a peptide. But again, you would still have a mechanism by which you could add certain things here using a directed method, because you would need to make sure it's directed, because otherwise you get adducts all over the molecule. So for non-peptide-based combinatorial libraries, things do tend to get a bit more complicated because you'd need to use quite specific chemistries. So that gives you an idea of the sort of chemistries. I've already talked about how you'd physically do it. So one of these approaches, one of the approaches that can be used is called the teabag approach. So the teabag approach was developed in the 80s. And the idea here is that you want to do reactions for similar chemistries in the same place at the same time. So the idea was to not organize them just by bead, but put the beads in a small mesh bag. So the beads would be placed in a labeled mesh bags, and this would be typically sort of 2 by 1.5 centimeters. The meshwork would be too small to allow the beads to get out, but the solvents and the reagents would be able to get in, so exactly like a tea bag. The bags would be placed in the solutions containing the first amino acid, so you generate your first amino acid of the peptide chain. The coupling reaction would take place, and then the big advantage of this would be that you then take all the bags out of their first reactions. So you could imagine if you've got 20 amino acids as your first amino acid, you'd have 20 pots for the first reaction, then you take all the bags and you put them into one pot to wash, then all to deprotect, and then you could sort them again and put them in our array of our 20 peptides for the second, 20 amino acids for the second amino acid of the peptide. So the big advantage here is that the washing steps and the deprotection steps are common, so you're saving time, you're saving money, and you're saving space. The end result of the teabag method is each bag will contain a single sequence. So it doesn't have to, they don't all have to be the same sequence, so you could have 1,000 different bags, and you could put them in different amino acids in different combinations in order to generate 1,000 different sequences. It would be quite labor intensive, but it would be possible. So the teabag method can be used to generate specific peptide sequences on a variety of different beads all at the same time. But this would be a method to generate specific sequences and generate one sequence per bead. Sorry, one sequence per teabag. However, if you wanted to do a completely random approach, if you wanted to do a combinatorial library where you were looking at random combinations of different moieties, different adducts, different amino acids, if you're making a peptide library, then you can use the mix and split approach. So the mix and split approach is similar to the teabag approach, except there is a lot more randomness to it. So you would still have your different pots. So if you had your 20 amino acids, you'd have 20 pots, one for each amino acid. I've just shown 3 here. So amino acid A, B, and C. And you would have your beads, and you'd split the beads evenly between the 3 pots. So in pot A, you'd generate beads druggitized with amino acid A, in pot B with amino acid B, and pot C with amino acid C. You would then remove the excess, you would wash, you would deprotect, and then you mix them all together, and then split them into 3 again. So you would effectively randomize at this stage. And then you split them into A, B, and C, and hopefully we can see that in A, we add amino acid A, in B, we add amino acid B, and in C, we add amino acid C. And then we go through the cycle again, and we'd mix and we'd split, and so therefore we'd add A, B, and C in the 3rd position. Now we can see by the end of just these 3 cycles and just 3 amino acids, we've generated 27 different combinations. If we extended this out to 20 amino acids and we did more steps, we would generate a huge variability, a huge combination of different peptides, hopefully we can see. So this is a good way of generating a lot of chemical diversity in a relatively short time. And then, because we have combinations of different chemicals at the end, we would then screen using our mixtures. So we would screen with the product of pot number 1 or pot number 2 or pot number 3. Now the big disadvantage here, of course, is that in our example here, each of our pots at the end have got 9 different chemicals in, so you'd have to work out what that was, and this is done through something called deconvolution. So the idea of deconvolution is, if we take pot number 1, which of these 9 molecules is the one giving rise to our effect? Which is the drug that actually works? So the key to deconvolution is that we take some of the mixture after each step. So, with recursive deconvolution, we know what our active pool is. So let's say our active pool is reaction number 2. But we need to know which of these 9 chemicals in the mixture is actually active. So what we do is we take a look to see what of the structure do we already know about. So we already know, because we're in pot number 2, the last residue is B. So our structure is XXB, where X is something we don't know and B is the thing we do know. So we know that the last amino acid is B. So what we do is we take the round 2 mixtures, so these are the mixtures with 2 amino acids, and then we add B to each of those. So we take the mixture from pot 1, the mixture from pot 2, the mixture from pot 3, and then we add B to each of them. And then we screen which of those is the active one. And from this, we can see that the active one is here in pot number 1. So we know therefore that the active compound has got X at the beginning, so it's A, B, or C at the beginning, and then A and B. And finally, we take our round 1 mixtures of A, B, and C, and add A, B to each one, and then rescreen again. So this recursive deconvolution allows us to go to our mixture, our complex mixture, and work out what is actually the active compound within it. So just to finish off, the idea of the mix and split approach is especially useful, especially handy during drug development, because the library size is enormous. So for a peptide library, each amino acid is 1 of 20, so for an octopeptide we can generate 25 billion combinations of different peptides. And it's now usual to test the compounds for biological activity as a mixture in solution following cleavage from the solid phase. So in each of these cases, we would take a combination of beads, we would take the peptide off, and then we would do something with it. Okay, so we've run out of time now, so the rest of this stuff is on Panopto from last year, so there are links on the Canvas page, so I'd recommend you have a look at that, please. The next session I'm at is Thursday morning, and so if you've got any questions from this material, please come and talk to me on Thursday.